We did one. We did neutral fats and triglycerides. Now we need to talk about phospholipids. Okay, so I'm just going to use this picture instead of me drawing one. If we take this and we get rid of a fatty acid, and we slap a phosphate group on it. So now I'm attaching this, which is going to contain a phosphorus, and it'll have some oxygen. I don't exactly know what a phosphate group looks like. I can't remember, but it doesn't matter. The whole point is a phospholipid is a triglyceride that has one of its fatty acids removed, and it's replaced with this phosphate group. Okay? So this is going to change the how it interacts with water. So before we had our lipids, our triglycerides, they're non-polar, they're not interacting with water. But now we have this phosphate group which has a charge, and so since it's charged, this is a polar head, we call it. So this polar head likes water, so we call it hydrophilic. These tails, they're non-polar, NP, and we call these hydrophobic. Philic, if you think of pedophilia, that is an attraction to children. Ped means kids. Philia is an attraction. If you think of organic chemistry, electrophile, that's an attraction to electrons. Nucleophile. There's a bunch of different examples. So phile means liking or loving, so this is water liking, and then phobia is a fear, so nonpolar are hydrophobic, so they're water fearing. Let's see what that means in terms of our orientation in solution. So if we remove that fatty acid tail from the triglyceride, we remove it with the phosphate group, it's a charged particle, it becomes, since it's a charged region on this molecule, we now have a polar molecule. We always talk about this phospholipid bilayer of cells. So this is my cell. And if I really look at this plasma membrane, then this plasma membrane is a bilayer, meaning two layers. And then if I zoom up on it and look at just this, then I'd have these two lines. But then if I zoomed up on it and really looked at it, then I'd see all these different phospholipids. Give me a sec. Let me just do this. Okay, so now we've zoomed in on our phospholipids. Let's look at the structure one more time. We have this polar charged head, making this a polar molecule that's hydrophilic, and these nonpolar hydrophobic tails. So when we're orienting this, if we're saying that this is the cytoplasm, and we're saying that this is called interstitial fluid, but right now we'll just say external fluid, so fluid outside the cell. And I do this same division where I say this is going to be the cytoplasm side of the plasma membrane, and this will be my fluid side of the plasma membrane. If you look at how these heads are oriented, these polar heads, these polarly charged phosphate heads are oriented towards the liquid. So cytoplasm has the cytosol on it. That's the liquid portion of this gel-like fluid in the cell. And then outside your cell, all your cells are surrounded by fluid. So these phospholipids orient in the correct way in order to make sure that their non-polar heads face each other. Right here. And there. And then these polar heads are facing the water. So those are phospholipids. They make up all these different bilayers, they make up all these different membranes. So the cell membrane, they make up the nuclear membrane, they make up the mitochondrial membrane. Is there anything else you want to say? No. So for now, that's all we care about. Hydrophilic heads can interact with water. Hydrophobic tails do not interact with water. Alright, next we'll do a steroid. I'll get a picture of a steroid really quick. So this is the steroid structure. Steroid structure is 4 interlocking carbon chains. Let's highlight them. 1, 2, 3, 4. So all these steroids, aldosterone, testosterone, cortisol, progesterone, they're all these 4 interlocking carbon chains. So these are going to act as fat-soluble lipids. Fat-soluble means it can be dissolved in something nonpolar, but it also means that it can pass through this plasma membrane. So fat-soluble or lipid-soluble material can pass straight through this plasma membrane. So it can just go scoot on through and make it inside the cell. If I was a polar molecule, I would need something like this integral protein that has a channel for me to go through. But since I'm a steroid, since I'm lipid soluble or fat soluble, I can pass straight through this plasma membrane. Steroids travel over large parts of the body, so they're produced in one part of the body and then act in another part of the body, versus eicosanoids. So eicosanoids, these are, I think 20, check real quick. So yeah, an eicosanoid is a 20 carbon biologically active lipid. So they work like steroids, so they're going to be able to pass through a plasma membrane and affect the cell's activity, but they don't work over long distances. They act locally, they're produced in one part of the body, and they affect things locally. So one example are prostaglandins. We'll talk about this a lot next semester when we get to inflammation. So we'll keep it at that for now. Eicosanoids, biologically active lipid molecule that acts over short distances.